Good afternoon, everybody. Today we'll be using a leaf porometer, which measures the stomatal conductance of leaves on this oak tree here, this Quercus virginiana. So what a porometer does is it measures the amount of CO2 going through this inner tube here. At this point, Torolf will show a diagram of the model in the with the lecture. But basically what we're doing is we're measuring temperature differences between the bottom of this instrument here and the top of this instrument here with some couple of electric resistors. It's a really simple machine to use, and it's relatively easy to use too. All that we really do is we find the leaf, right? So the idea that we want to show is that right now we have sunlight hitting this part of the leaves, which are going to have higher photosynthetic activity. Also, another thing to think about is the time of day. Right now it is approximately 6:35 p.m. in a summer month, therefore we're getting closer to sunset. At this point, even though these are shrubs, these are trees, the conditions are favorable due to the low vapor pressure deficit that exists at this time of the day. So right now we actually expect to see higher transpiration rates from trees than, say, for example, 12 or midday. So all that we have to do again is we have our clip after it's been calibrated, and we go ahead and attach it to our leaf here. So we measure, our instrument starts recording. The test takes about 30 seconds to do. So the test is done. We could take various samples and we could take the average of 3 readings. So right now this has been my 3rd sample now, and I have between my 3 previous readings I have an average of approximately 600 macromoles per meter squared per second of CO2 leaving this leaf. So this is the the sunlit part of the tree. Now we're going to go further inside here, or a little taller over here, and we're going to try to capture the differences between the sunlit portion and the shaded portion. It might be higher in the shaded portion, it might be lower. So we're going to do that and find out. Alright, everybody, so. Alright, everybody, so now I'm under the shade. This part of the canopy is under the shade, right? So it's not getting direct radiation. It's getting diffused radiation. So what we have here now, again, we're going to take the test 3 times. And again, the test takes 30 seconds. We'll get those measurements and we'll see if it's lower or if it's higher. So we'll commence the test now. Alright, so after 3 tests, what we saw is we have a lower stomatal conductance. The average of our 3 tests in the shaded part of the region was approximately 330 macromoles compared to our sunlit region that was 600 micromoles per meter squared per second. Therefore, at this time of the day, when we have low vapor pressure deficit, when it's closer to sunset, the sunlit part is photosynthesizing more, which means, in a sense, that the stomata of these leaves are more open than the ones in the shade.